Hello everyone, and welcome to my Capstone project where I will describe for you a bacterial transformation activity. The summary of the activity is that students will construct paper recombinant plasmids to simulate the methods genetic engineers use to create modified bacteria. They'll learn about enzymes, DNA, and genes and their roles in the modification of organisms. And in this particular model, they'll isolate a mammal insulin gene and combine it with a bacterial gene sequence, a plasmid, for production of the protein insulin. This activity, I really liked it as it is, and I kind of thought about how I would modify it for my class. I think that it could use a bit more inquiry, and so I would like to do that by introducing the activity with an open-ended segment where they research genetically modified organisms. I could give them some ideas, glowfish, or they could even analyze the foods they ate that day and try to see which are genetically modified and for what purpose. Then they would report out to the class about those foods or other examples of genetically modified organisms, and that could kind of be a springboard for a discussion about GMOs. And then we would do this paper activity, and I feel like we could then extend the activity, especially for advanced classes, by performing an actual transformation of E. coli by incorporating a fluorescence gene and an antibiotic resistance gene into the E. coli. And as I said, that's more of an advanced lab, but it could be part of a sequence. I feel like this activity aligns with the STEM practices listed on this slide, specifically asking questions and defining problems and using models. In terms of science inquiry, this is where I felt the activity may be lacking, although I still feel it has value to help kind of bring an abstract concept to life for the students. And they are engaging in a scientifically oriented question. And if we discuss GMOs as kind of a wrap-up, students could take a position on how whether they feel that this type of technology is beneficial or whether it has some risks, and that would be communicating and justifying explanations. I included the inquiry continuum here, and you can see that I feel like this activity specifically would be, pretty much exclusively in the teacher-guided sections, this first column that I'm highlighting here. Okay, so I took some photos of this activity. My kids went away and my students are taking finals, so it was just me doing it. But the first slide here is showing you the materials that are needed. There are two sequences of DNA. The white one is the plasmid, and the gray one is the gene, the insulin gene. This second slide is showing you the plasmid after I've taped it together, because the plasmid is a circular piece of DNA, so that's what it would look like. I would probably have copied these on two different color paper to make it a little bit more of a contrast for the kids, but I forgot to do that. In this slide here, you can see that I highlighted and then drew a line where the restriction enzyme would recognize a sequence in the DNA of both the gene and the plasmid, and that's where that enzyme would cut the DNA. And so I'm showing you the cut in the plasmid and in the gene. And then I'll kind of go back and show you this, what you're seeing is a staggered cut, and they're called sticky ends when the cut is staggered like this. So then this gene could attach and be incorporated into this plasmid. So with little pieces of tape, the kids can incorporate that insulin gene, and then what they see is this recombinant plasmid at the end that includes the gene for human insulin. It would then be incorporated into the bacterial cell, and the bacteria could act as a little insulin-making machine. Cool activity. I really enjoyed working with you all in this class. Good luck and have a great summer.